And start if you're ready. Typically with these meetings, I begin with a land acknowledgement and then I introduce the speaker. And then I can hand it over to you. So if you're all set, I'll get started with that.
Yeah,
Great.
I'm all set.
Okay, wonderful. Well, welcome everyone. We're thrilled to have you with us this evening. I'm Chris Watts. I'm the Vice President of the Grand River chapter of the Ontario Archaeological Society. Tonight we have a guest speaker, Professor Karine Taché from the University of Laval. So I'll just begin tonight with our land acknowledgement before introducing our speaker. So the OAS Grand River chapter acknowledges that much of our work takes place on the traditional territory of the Arawandran or Neutral, Anishinaabe, and Haudenosaunee peoples. Our chapter is located in the Haldeman Tract. That's land granted to the Six Nations that includes 6 miles on either side of the Grand River in southern Ontario. And our active work towards reconciliation takes place across various chapters of the OAS, but for us through various research, learning, teaching, and community building initiatives. And a lot of this work is centralized within the main Ontario Archaeological Society office in Toronto. So with that, I'd like to introduce our speaker for this evening, Professor Karine Taché. It's an enormous pleasure to welcome her this evening. Professor Taché is an assistant professor in the Department of Historical Sciences and Canada Research Chair in Biomolecular Archaeology at the University of Laval in Quebec. She completed her PhD at Simon Fraser University in British Columbia and is a specialist in the archaeology of the Northeast. Indeed, many of us know her perhaps best from her work on meadowwood trade and exchange practices. I was just commenting earlier that many of my students often write on this topic and cite Karine endlessly in their papers. So I've been meaning to have her give a talk for some time now. Prior to joining the faculty at Laval, she was an assistant professor in the Department of Anthropology at the City University of New York, CUNY, Queens College. One of the unifying themes of Professor Taché's research is food and the various cultural practices that surround its production and use. So to this end, she has developed techniques in analytical chemistry that serve to sort of extract and identify organic materials, molecules, preserved in archaeological contexts, especially within the sort of porous matrices of various sort of ceramic fabrics. Professor Taché conducts research in the Laurentian Highlands region of Quebec, and just now was telling me about another field school she's doing in Quebec City or near Quebec City, where one of her ongoing research projects, at least in the Laurentian Highlands, focuses on the way in which pottery is used in the Quebec subarctic. And through a grant from the Canadian Foundation for Innovation, she founded the Laboratory of Biomolecular Archaeology at Laval, one of Canada's largest and leading research centers in archaeology and archaeometry. Her approaches contribute to the development of new methodologies applied to the study of organic residues in archaeology and to the enrichment of our archaeological interpretations, in particular, those relating to Indigenous and European material culture traditions in North America. So with that said, I will turn the floor over to Professor Taché.
Good evening, and thank you very much, Chris, for that introduction. And thanks for inviting me to present at this series of conference organized by the OAS Grand River chapter. It's always a pleasure to share my ideas and discuss with colleagues, so don't hesitate to ask questions, provide comments during or after my talk, I don't know, as you wish. Before I start, I also want to acknowledge that my research involves material and sites belonging to the ancestors of several First Nations that are still around today, and to whom I am grateful for authorizing me to do my research and for collaborating with me on that research as well. So in this talk, I will try to show how the study of potsherds dating to the Woodland period in northeastern North America can contribute to the study of foodways. So this potential, this heuristic potential of pottery has somewhat been overlooked at the benefit of morphostylistic analysis. So we've been more interested in the container than in the content. There's a good reason for this, because until recently, archaeologists simply did not have access to the contents of these containers, of these ceramic containers. But in recent decades, the development of new biomolecular technologies, applied to material culture has made possible the identification of a range of perishable products that were formerly inaccessible to archaeologists. So it's in this context that I proposed in 2020, as I was saying earlier to Chris, the creation of a laboratory at Université Laval equipped for the extraction and GCMS characterization of lipid residues preserved in archaeological contexts. That proposal was accepted, and 4 years later, believe me, I did not know what I was getting myself into. The lab is now functional since a few days ago. The lab and the Canada Research Chair was set up to accomplish a number of objectives. I've kind of summarized them here. So extract and characterize lipid residues preserved in archaeological context, develop new methodologies that we can apply to the study of organic residues in archaeology, and ultimately enrich our interpretations of the past, particularly those relating to the study of foods and foodways. Because knowing what was placed in pottery provides strong evidence for how and why it was used. This is kind of the rationale for many studies involving lipid analysis of ancient ceramics. Identification of vessel contents has been conducted at various spatial-temporal scales, from the identification of household or site-specific patterns in pottery use, to the documentation of variability between pottery types, and the investigation of broader scale events such as pottery adoption or transition to farming. So the first project that I want to talk about today involves a broad scale event. So I was looking for my postdoc after my PhD on the Middlewood interaction sphere at pottery adoption in the Northeast. So I conducted this research in the BioArc group in the UK at the University of York, also as a member of the Early Pottery Research Group. The Early Pottery Research Group is an international and multidisciplinary group of researchers working within a network of collaborations to study the origins, the use, the dating, and the social context of early ceramic containers, but mainly pre-agricultural containers in different regions of the world, as you can see on this map here. So that's it's within that network of collaborations that my work was conducted. And the idea behind such comparisons was to identify common drivers, but also regional specificities in the initial uses of ceramic vessels by hunter, fisher, gatherers worldwide. So my research looked at pottery sherds, typically referred to as Vinet 1 pots, after the site in New York State, where William Ritchie first defined that type in the 1940s, based on a list of attributes related to pottery paste, surface finish, morphology, and so on, and yeah, mainly. And then I selected Vinet 1 potsherds from approximately 135 distinct vessels, or a minimum of 135 vessels from over 30 sites. So of course, encompassing a wide range of environmental and cultural context. So my work with these early pottery sherds has revolved around the recovery and analysis of organic residue. Organic residue analysis employs complex analytical techniques developed in analytical chemistry to identify the nature and origins of organic remains that simply cannot be characterized by more traditional recovery techniques, simply because residues are amorphous and invisible. So you can't pick them in a screen or in a flotation device. You need to apply GCMS analysis and other type of techniques. These techniques have been applied to a wide range of materials, some of which are shown here. So sediments, coprolites, even stone and metal tools, but most progress has been made in relation to the analysis of lipids from ceramic cooking pots. And that's mainly what I'm going to be talking about today. Now, how do we get lipids from pots? There are mainly two types of residues. So food crust, visible residues. If such visible residues are present, we need only a few milligrams that can be scraped from the surface of a sherd and then analyzed. But fortunately, potsherds lacking food crust can also yield a lot of information. So that information is trapped in their paste because lipids are easily absorbed within the porous paste of ceramic containers during cooking. For absorbed residue, we need approximately 1 gram of sherd powder, and this powder is obtained by drilling the sherd with a Dremel type tool. So it's destructive analysis. We're trying to develop methods so that it becomes less destructive. So now we're using less than 1 gram, as opposed to approximately 2 grams when I started doing my research. So lipids that are trapped in food crust or in the ceramic matrix can then be extracted by the addition of organic solvent. And there's established procedures in the lab that lead to the extraction of lipids, and then they can be analyzed through a variety of analytical techniques. I'm not going to be explaining in details these techniques, but if you have questions later on, I'll be happy to answer them. Lipids have been shown to survive better than other classes of organic materials over considerable time periods, so they can be used to a certain extent, at least, to tie chemical signatures to food products. But some classes of lipids have better diagnostic values than others. Fatty acids are found in every living organism, so they do not have good diagnostic value. They are found in various proportions, but using proportions and ratios is somewhat difficult because of all the degradation processes that we need to really control if we're going to be using ratios. So we mainly rely on chemical compounds that are called biomarkers, and that can be used to identify quite precisely a range of resources. Just a few examples are shown here: birch bark products, pine resin, beeswax, aquatic resources, and there's many other biomarkers that are that have been identified and that are very useful to confidently identify a substance and then document the use of that organic the use of that product in the past. Now, it's important to combine analytical techniques to get the most out of those lipids. So my work revolves around a combination of techniques that I've listed here. So isotope and molecular characterization. So bulk carbon and nitrogen isotope analysis, GCMS analysis, and compound-specific stable isotope analysis. Again, I'm not going to go in detail about these techniques, but I'll show some results. And if you have questions about how these function, don't hesitate to ask questions later on. So going back to my research on Vinet 1 pottery, this combination of techniques led me to show that among early pottery samples that I analyzed, a majority produced lipid profiles typical of degraded aquatic oils. Here you can see a chromatogram, so a profile that is typical of degraded aquatic oil, based mainly on a range of biomarkers that meet the established criteria for the identification of freshwater and marine resources. So 75 samples had such profile. By contrast, only a minority of samples pointed to animal fat or plant oil as main sources of lipids. And what I'm showing here on the right side of the slide is the fact that lipids in that study at least tended to preserve better in the ceramic powder, in the matrix, rather than in the food crust. And this is a pattern that I'm maybe not as clearly as this here, but that is repeating itself in my other work as well, the one that the other studies that I'm going to be talking about today. So that was interesting to see that that predominance of aquatic resources. What was surprising also was, and this is the result of single compound isotope analysis, was that the evidence for ruminant lipids was surprisingly limited. So here is the ellipse here was built according to reference values coming from wild ruminants. So very little samples had values that we could link to ruminants. And this was surprising given on the one side, the abundance of deer and elk in the woodland environments of northeastern North America at that time, and also the abundance of white-tailed deer in faunal remains associated with many early woodland sites. So this mismatch between the evidence from the analysis of food remains on the one hand and organic residue data on the other suggested to me that there was a preference for the processing of aquatic resources in pot that was not simply a reflection of the wider subsistence strategy at the time. And combining the results of that analysis with botanical and faunal data, as well as evidence for new social developments that I analyzed as part of my PhD associated with the early woodland period, I suggested that the invention and widespread uptake of pottery in northeastern North America was associated with the need to prepare aquatic resources in the context of increased social interaction at that time. Episodic gatherings could have been driven by short-lived availability or abundance of certain aquatic species during the spawning season, for example. And of course, at these occasions, cooperative harvesting of aquatic resources may have taken place in addition to trade and a range of other activities that have been documented in the archaeological record. Now, this said, we have to remember the relatively small number of Vinet 1 vessel at any one site where this type has been documented. And this is still intriguing. I do not have an explanation for that at the moment. It's still a question for future work, and I'm happy to discuss that as well with you. But still, I think we can agree that this research on early woodland pottery switched the focus from traditional typological approaches to kind of investigate and learn more about the actual uses of these cooking instruments. So that was my first study that I wanted to share briefly with you. The one that I did as a postdoc at the University of York. Now, turning to site-specific patterns in pottery use, and turning also to how the use of clay containers changed after the adoption of agriculture by several First Nation communities in the late woodland period. This is a second case study that I want to talk about today. This research is ongoing, but I'll be talking about, because I want to kind of analyze late woodland pottery from many other sites, but I'm going to focus on two sites, the Dawson and Ising site today. Because these are already revealing interesting site-specific patterns, which again would probably have gone unnoticed without organic residue analysis, lipid analysis more specifically. So a few words about the Dawson site to begin with. So it's the Dawson site, for those who don't know, is an Iroquoian village site discovered in the 19th century, 1859, by construction workers who unexpectedly discovered material remains and features in downtown Montreal. And Dawson has long been a favored candidate in the search for the famous village of Hochelaga, encountered by Jacques Cartier during his second trip to the St. Lawrence Valley in 1535, although this hypothesis has never been proven. As one of the first Iroquoian sites ever described, the Dawson collection became seminal in the history of Northern Iroquoian archaeology. But it was only properly studied in the late 1960s by Bruce Trigger and Jim Pendergast. And you can see the book here. But at that time, still in the 1960s, the nature of the data recovered in kind of an antiquarian manner characteristic of that time has been a limit to interpretations. Fortunately, in 2016, the firm Ethnoscope Inc., in collaboration with the City of Montreal, conducted salvage archaeological excavations at the Dawson site. And this led to the recovery of new data, including pottery sherds from approximately over 89 and perhaps over 89 ceramic vessels. Now, after a gap of more than a century and a half, the Dawson site was suddenly revealing some new and now rigorously excavated material to work with. And that's where I was contacted to conduct state-of-the-art analytical methods. So lipid analysis was conducted to new data from that old site. So we selected 43 samples representing a minimum of 25 vessel units. These were submitted to the same analytical techniques mentioned previously. So we did both molecular and isotopic characterization. And the most distinctive molecular feature of those sherd, here again, is the presence of biomarkers for aquatic product for aquatic products in up to 89% of the interpretable vessel units. So all the vessels that had enough lipids to be interpreted. So clearly, aquatic resources, fish, were a favorite ingredient among the Dawson cooks. But the question was, were fish cooked alone in these containers, or were they mixed with other foods? And it's only through isotopic analysis that we were able to kind of favor the second scenario. So we did both bulk carbon and nitrogen analysis and single compound isotope analysis. Now, this graph here shows the carbon and nitrogen isotope values obtained on 10 food crust sample from the Dawson site in red here, and they are put side by side. They are compared with the values that were obtained on the Vinet 1 pots described earlier or discussed earlier. So as you can see, there is no overlap at all between the blue and the red dots, and this is because maize contributed to the Dawson residues. So you can see that the Dawson residues in red are enriched in carbon, which is characteristic of C4 as opposed to C3 plants. And because maize is by far the most common plant with a C4 photosynthetic pathway in pre-contact northeastern North America, these more positive values can be used to estimate the importance of maize or to attest to the importance of maize in those spots. The contribution of terrestrial animal fats to a minority of Dawson pots was also shown. Here you can see the chromatogram, so molecular characterization of one such pottery sherd that yielded a profile typical of animal fat. So you can see the abundance of C18 or stearic fatty acid. The predominance of stearic over palmitic fatty acid is often taken to infer the presence of animal fat. And this is also supported by single compound isotope analysis. This graph shows such analysis here, where you can see that quite a few pots from Dawson appear to be found within that ellipse corresponding to wild ruminants. So what we seem to have at Dawson is a mixture of resources, but can we be more precise than that? So we have fish, we have maize, and we have also in a few pots potentially ruminant fat. Can we be more precise than that? Well, characterizing mixture of food is a great challenge in organic residue analysis, but there are ways we can try to kind of to do that. So one is to use Bayesian mixing model. This is one Bayesian mixing model that was generated by an open source software that is named Fruits. So I was kind of putting isotope values and reference values in the model, telling the model to estimate the relative importance of aquatic oil, maize, and ruminant fat in the different pots that I analyzed. So the result is shown here, and quite interestingly, it suggests that maize was present in all the sample and that it was dominant in at least 40% of the potsherds analyzed, despite being completely invisible in the chromatographic profiles. So again, this begs for the combination of analytical technique, because otherwise the presence of maize would have gone completely unnoticed. So as you know, probably ethnohistorical records contain a lot of mention of cooking recipes where maize, fish, and pieces of dried meat, sometimes berries as well, are cooked together. The most notable and the most frequently mentioned such recipe is the famous sagamité. Well, it could well be that the results obtained at the Dawson site show that this mixture, that this dish was already part of the culinary tradition of the people inhabiting the site. So it's coherent with such a recipe. I'm going to stop here for Dawson, but you're welcome to ask questions later. I'm going to turn now to the second site, the second late woodland site in the St. Lawrence Valley, the Heising site. It's another Iroquoian village site. And here we have so far analyzed 23 samples that we submitted to lipid analysis. What's interesting at the Heising site is that we have completely different chromatographic profiles. So here you have 18 out of the 23 samples and 22 samples that were interpretable, I should say. One didn't have enough lipids. So 18 out of 22 that had lipid profiles characteristic of plant oil. Only 4 had aquatic biomarkers. So this is quite different from Dawson. And these data, well, right away suggest maybe that the pots from Heising's were specialized cooking implements used for the preparation of plant foods, a pattern that is completely different from Dawson once again. And we also submitted those pottery sherds to single compound isotope analysis. We didn't do carbon and nitrogen because we only had absorbed residues, not food crust. But single compound isotope analysis suggests that the plant, so the Heising site values are in red and the blue ones are from Dawson. So it suggests here a C3 rather than a C4 signature. The blue pots here from Dawson suggest a mixture. So when you have both maize and fish, obviously you'll have intermediate isotope values. But the values from Heising's are lower in the graph, suggesting more of a C3 plant signature than a C4. This is surprising given that we know that maize was cultivated at that site. And this is ongoing work. Further investigation is needed to interpret these results. But one possibility is that, well, one possibility is that they were cooking C3 plant. But another possibility is that we are dealing with maize, but in a different form. John Hart and colleagues had showed and published work showing that certain types of, certain form of maize, whole kernels, whole grain hominy, cooked in ceramics were largely masked by C3 resources as opposed to hominy meal. So it could be that different isotope signatures are to be expected if, rather than sagamité, the pots from Heising's were involved in the transformation of ripe corn into hominy corn by boiling into a mixture of water and hardwood ashes, as this is a method that was described in ethnohistorical documents. But this is something for future work. These are hypotheses that I want to be testing in the future, in parallel to doing more, to analyzing more samples. But you can already see that through lipid analysis, we can really see site-specific patterns in pottery use here during the late woodland period. So I'm going now to be turning to yet another project, the project in the High Laurentian that I was mentioning earlier, that I was talking to Chris about. So results from the St. Lawrence Valley, where Iroquois nations lived as semi-sedentary agriculturalists in the late woodland, can be compared with residue data from more northerly regions in the Canadian Shield, where the hunting, fishing, gathering way of life continued until the contact period, essentially. And one such region where I've been conducting work is the High Laurentian. I've been doing this work in collaboration with the Anishinaabe First Nation community. Here you see the Grand Lac Nomineng, where my team and I have been searching for a while, excavating for a while. And I want to say that this region is part of the ancestral territory of the Algonquin Anishinaabe First Nation. And I'm taking this opportunity again to thank them for their collaboration. This research would be impossible without their participation and their support. So surveys and excavations on the shores of Grand Lac Nomineng have been taking place since 2015. Here you can see a team of archaeologists excavating with our Anishinaabe partners, with Université Laval students, and local volunteers who have the archaeology of their region at heart. These excavations and other nearby have revealed an entanglement of campfires, testifying to multiple reoccupations at this location for more than 5,000 years. And associated with these heart features are, as we can expect, thousands of burned faunal bone fragments and a lot of ceramic potsherds, like this one here on the right, used for cooking. In total, since 2015-16, it's approximately 4,000 potsherds, representing at least 34 distinct containers that have been discovered. This is considerable, given that no ceramic had been documented there before we started our research. Had been documented in the High Laurentian, but not so surprising at the same time, because there are several other regions in the Canadian Shield connected to the High Laurentian by the hydrographic network that had already yielded ceramics. So we are finding more and more pottery in these more northerly region. And as you can see at this site around on the bank of Grand Lac Nomineng, the pots have been assigned to early, late middle, and late woodland period. So no early woodland potsherds so far. So among those 34 vessel units identified, 23 were subjected to the same biomolecular analysis mentioned before. And again, here you can see a graph showing the result of single compound isotope analysis, where we get the values, the isotope values for C16 and C18 fatty acids, and the results of the pottery from the High Laurentian are shown in red here, and they are compared with the blue circles are values that were obtained on the early woodland pots discussed at the very beginning of this talk. So again, you can see a pattern that is different in the High Laurentian compared to what was found before. And what is interesting is that we have quite a few samples that actually fit in that ellipse here, in that circle, representing terrestrial ruminants. And sorry, I just noticed that I didn't translate that graph. So terrestrial ruminant here. So it's interesting because not only does it contrast with what we found for previous period, but it could reveal a way of using ceramic containers that is specific to more northerly region where wild game, wild moose, wild deer still occupy an important place in dietary practices today. So these results are quite interesting. Again, we turned from more typical typological analysis to investigating the use of ceramic containers, but I am at the point of my talk where I have to admit that lipid analysis cannot answer all our questions. There are a lot of challenges and limitations. One of them being that lipids, as opposed to protein and DNA, I'm sorry, cannot precisely identify the resources, so we cannot or only rarely identify a resource to the species. We can only identify, we must stick to broad categories: ruminant, non-ruminant, aquatic, plant, C3, C4 plants, but we cannot go more precise than that. So this is one limitation. Another big challenge is, as mentioned earlier, is the characterization of mixtures. A lot of time when we have species that are poor in lipids, like plants, for example, that are mixed with species that are rich in lipids, like animal or fish, the plants or lipid poor resources tend to be represented or masked. So it's not always easy to see these plants with lipid analysis, despite the fact that isotope analysis can sometimes reveal them. Another limitation is the fact that when we do identify several resources, it's difficult to know whether these were mixed together in the pot as a single cooking events or as successive cooking events. So, and we know that the basis of our culinary traditions are almost always made up of a complex mixtures of several ingredients. So it's to this point, it's very difficult with only lipid analysis to reconstruct recipes. So what do we do in face of these challenges? Well, to overcome some of those, my collaborators and I imagine a research project on foods and foodways in the High Laurentian region that combines biomolecular archaeology, biomolecular analysis with experimental archaeology and indigenous knowledge. This is a 5-year research project. We're already in the 3rd year, and we have already carried out quite a few experiments that are already helping us better understand the role of ceramic vessels in culinary practices. The first year of that project, our main objective was actually to test the quality of local clay for making ceramics. So making pots. So we prospected the banks of the Lièvre River where clay deposits had been observed during previous research. We collected clay from different sources, tested them with different types of temper and different proportions also of temper before drying these small pots and cooking them over open fires. When it was shown that this clay could be used to make pots, we then continued and produced larger containers like this one here that we tried to also, we also tried to reproduce the decorative motifs that we observed in some of our pots. And last summer, we've put some of those containers to the test for the first time to prepare different recipes inspired by Anishinaabe culinary practices. And for this component of the research, we are very lucky to be partnered with Chef Susan Nottaway, an Anishinaabe chef, who has an intimate knowledge of her territory, but also of the recipes of her people, a knowledge that has been results from gestures, from practices, from traditions passed down from generation to generation. So because our biomolecular analysis had shown that terrestrial ruminants were dominant or were an important content in those pots, moose and deer game were central ingredients in Susan's cooking last summer. So she prepared moose stomach, moose nose, moose kidneys, and several stews involving deer and moose game. In addition to other recipes, and here you can see kind of bear fat popcorn. So when you do prepare bear oil, you divide the liquid from the solid part, and this is the solid part that you can eat just like popcorns. We also prepared fish and so on. And I want to also show that each of these ingredients were cooked individually, because eventually all these replicates will be analyzed using biomolecular techniques, and the results will be compared with the archaeological signatures obtained. So we want to know what the individual ingredients give and what the mixture give in terms of isotope and molecular signals. Some pots were used for a single recipe, other were used to make several recipes. So we're going to look at all this in the future in the lab. I will conclude soon, but I want to point out that no ingredients were wasted. So all the recipes were eaten, were consumed. The approval of Susan's father, Salomon, was always an anticipated moment. And because we want the benefits of this project to go not only to the scientific community, but also to our partners from Kitzigan Zibi, we have also planned a documentary that is being done by Dulce Menes that you see here, in addition to